Good afternoon, Namaste to all dear learners. I, Dr. Mahesh Singh, Professor Plant Pathology, College of Agricultural Sciences, Tirthankar Mahabir University, Muradabad, Uttar Pradesh. Welcome once again in NIOS live studio program. Dear learners, hope you all are doing very well. Today is a very important topic we will discuss, that is diseases of pulse crop basically and their integrated management. We will discuss about their integrated management for food security. Dear learners, nowadays there are so many crops affected by the numbers of diseases as well as insect pests. So we will discuss in detail about the diseases and their causal agent, what are the symptoms affecting the crops, so that the learning objective for the end of the session you will be able to understand about the identifying the major diseases that affect the pulse crops and describe their causes, their symptoms, their spread of the diseases, as well as understand how the pulse diseases reduce yield and they affect the food supply also, and how the different methods that you can manage this kind of diseases, and applying basically integrated disease management practices for the better production of the crop, pulse crop, for enhancing the food security as well. Dear learners, as you know, the pulse belongs to the family Leguminaceae, that is also known as Fabaceae, and they are rich source in protein, that is basically essential amino acid, especially lysine, vitamins as well as minerals also. Pulses from an important part of the diet, especially in vegetarian person, that contributing the nutrition for food security, and they fix atmospheric nitrogen that improve the soil fertility, as well as they also helps to the crop rotation for the sustainable agriculture. It provide the income to the farmers and that support to the livelihood through the domestic use and export also. Dear learners, the importance of pulses as also it require less water, and it also improve the soil health by adding the organic matter, and their productivity is affected by the diseases like fungal, bacterial, viral, nematode. There are so many kinds of biotic as well as abiotic factors that responsible to cause diseases, and the insect pest also that leading to the significant yield loss. Dear learners, the proper disease and pest management is essential for the improving yield and ensuring the food as well as nutritional security. Dear learners, these are some list that is important for important pulse crop. Like you know about the chickpea, that is also known as gram or chana in Hindi, and the botanical name is Cicer aritianum. Next is important pulse is pigeon pea, that is arhar or tuar dal known in Hindi, and the botanical name is Cazinus kazan, like that lentil, that is masoor, garden pea, that is matar, Pisum sativum, and cow pea, lobia, horse gram, that is kulthi, and green gram, moong bean you can say in Hindi, and Vigna radiata in botanical name, and like black gram, urd, rajma, kidney bean also known as, and soya bean, and broad bean. Broad bean is known as in Hindi bakla, and cluster bean, that is guar in Hindi. So these are some important pulse crop basically, and apart from these there are so many other crops are also cultivated in so many part of the world, but these are the basic major crops which are growing all over the world. And dear learners, the there are so many challenges that affect the pulse crop, like major diseases fungal, bacterial, viral, nematode, and including this wilt. The major diseases is wilt, root rot, blight, rust disease, powdery mildew, and mosaic diseases. They causes basically 20 to 60% yield loss. It's depending upon the severity of the disease, but sometimes even more than that also. I mean, sometimes it causes up to 100% depending on the severity also. If the crop get affected in the early stage of the growing season, so it causes severe loss. It lead to the poor grain quality and reduce the market value also. Disease spread increases under the favorable weather condition, lack of resistant variety, and poor management problem. They can also worsen the problem also. And integrated disease management, that is IDM, is essential for the control of stable production of the crop. And just I discussing the major diseases like wilt, root rot, ascochyta blight, cercospora leaf spot, anthracnose, rust, powdery mildew, phytophthora blight, alternaria leaf spot, damping off. These are the basic major diseases which are responsible to cause more damage in the pulse crop. And some bacterial diseases like bacterial blight, bacterial leaf spot, and in viral diseases yellow mosaic virus, sterility mosaic virus, chickpea stunt, lenting yellowing virus, and cowpea yellow mosaic virus, like that bean leaf roll virus. These are the viral disease. Like that, so many other insect pests are also responsible for damaging the crop, like you have seen in pod borer, like gram pod borer, that is Helicoverpa armigera. It is a major pest of chickpea, pigeon pea, lentil, etc. They feeds the food as well as seed also. And aphid is another important insect pest that is responsible to cause severe damage in pulse crop. And white fly also, that is Vemisia tabaci, spotted pod borer, pod fly, pulse beetle, etc. And but pulse beetle is basically causes damage in storage condition. So these are the some insect pests also responsible to cause damage, create damage in the pulse crop. And we will discuss basically today in about the diseases of the pigeon pea and pulses also. So first we will discuss about the pigeon pea disease. The first disease is wilt disease of pigeon pea, and you know the distribution and economic impact, how they affect the crops. The wilt is one of the most common and destructive disease of pigeon pea in India and many other tropical countries. This disease is widely distributed in India, African country, and Southeast Asia, where the pigeon pea has been growing in as a major pulse crop. And in India, it prevalent in all pigeon pea growing states, especially in Uttar Pradesh, Madhya Pradesh, Maharashtra, and Andhra Pradesh also. And it is economically important because it causes the partial or complete plant death, resulting the 20 to 100% yield loss depending on the severity. The disease also reduce the seed quality, and it affect the germination percentage, leading to the loss of both yield as well as income for the farmers. And the symptoms, you know, basically the symptoms of this disease is yellowing and the dropping of the leaves, and plant get stunted and the lower part of the plant, and sudden wilting may happen of the branches as well as entire plants also. and when the stem is cut broken, and it you will find the basically dark brown patches under the stem, and the xylem vessels basically completely block, and the water and nutrient absorption can be checked clearly, and the partial wilting may occur in patches across the field, and in the severe cases the whole plant dry up before the maturity. So this is a basically fungal pathogen responsible to cause this disease, that is wilt of pigeon pea. The pathogen is Fusarium udum, and basically this pathogen get enter in the root of the pigeon pea plants and get damage the whole crop, and the fungus survive basically in the infected soil, plant debris as well as through the seed also. The infection occurs through the roots, particularly through the natural openings and wounds also. This disease is spread by means of rain splashes, irrigation water or contaminated soil also. Once it enter into the inside of the plants, fungus completely block or choke the xylem vessels, and it preventing the water transport, and that's why it cause or create wilting symptoms. And the favorable condition for this spread of the disease is warm temperature 25 to 30 degrees centigrade, and moderate soil moisture is favored to this disease development. And how you can manage this kind of disease, basically there should be the you have to apply the resistant variety and available in the market. Basically you have to grow resistant, either tolerant variety, for it is a most economical and eco-friendly method to minimize the disease severity. And there are so many like Bahar, Maruti, UPAS 120, ICPL 87119, so many varieties are available. So you can prefer the basically disease tolerant variety or disease resistant variety, you can choose as per your availability. And these variety exhibit good field tolerance to the Fusarium udum and perform well under the different agroclimatic condition. And next is the cultural practices. It is a very important to know about the cultural practices, how it responsible or how it can minimize the complete disease management process. So first of all, crop rotation, it is a very important to avoid the at least 3 to 4 years or more than that of the crop rotation with the non-host crop like cereal crops, maize, sorghum or wheat, or other oil seed crops like mustard or sunflower apart from the pulse crop. And you have to rotate basically. And the field sanitization, you have to uproot and destroy the infected plant and crop residues after harvesting to reduce the fungal survival in the soil. And the next point is soil solarization. Before sowing, you have to expose moist soil to strong sunlight for 3 to 4 weeks by covering it with the help of transparent polythene sheets, so that the moisture should be there, and the temperature below the polythene sheet get rise, basically more than 60 degrees centigrade, or that so that all pathogens get destroyed under the soil. And proper drainage system should be there. You have to avoid stagnant water in the field, so it favor the disease development. Sowing time, you have to also manage the or recommend the sowing time to avoid the condition which are favorable for the wilt incidents. And you have to also control the weeds surrounding the field, because it harbor the alternate host for the pathogens, dear learners. Next is a biological control. You have to also prefer the biological control system like seed treatment, Seed treatment with the help of Trichoderma hergianum or Trichoderma viridi at the 10 rate of the 10 gram per kilogram of the seed before sowing. The bioagent colonizes the root surface and it protects against the fungal infection. And soil application, you can mix the Trichoderma hergianum 2.5 kilogram per hectare with the 25 kilogram well decomposed FYM or compost you can also take, and you can broadcast it in the field 7 to 10 days before the sowing. And you can also use the rhizobium inoculation after the fungal or bioagent treatment. You can inoculate the seed with the recommended rhizobium culture for better and insulation of the plant vigors. And there is the last weapon. If all the techniques you are not getting the better result, you have to also apply the chemical method can be used along with the cultural as well as biological measures, especially in heavy infested areas. And you can treat the seed with the chemical fungicide like carbendazim or bebastine at the rate of 2 to 3 gram per kilogram of the seed before sowing. And alternatively, you can also use thiram or carbendazim at the ratio of the 2:1, and it can be used broad spectrum protection also. And soil drenching, in early infection cases you can drench the soil near the wilted plants with the carbendazim 0.1% solution to prevent the further spread of the pathogen. You can also avoid the excessive chemical use to maintain the soil microbial balance. So you have to be specific for the chemical use. Dear learners, the next disease is Ascochyta blight of the chickpea. Basically it causes the heavy loss in the chickpea plant, and Ascochyta blight is one of the most serious and destructive fungal disease of chickpea and other pulses as well, especially in cool and humid region. The disease affect leaves, stems and pods at the growth stages as well. It can also lead to complete crop failure under the favorable condition. It occurs in almost all chickpea growing areas of the world like India, Australia, Canada, USA, Ethiopia as well as West Asian part of the world. And in India it is most common in northern as well as central zones where the cool moist weather prevails during the crop growth. Dear learners, yield losses it range from the 10 to 100% depending on the disease severity and weather condition also. The disease reduces seed quality, germination percentage as well as market value that affect both farmers and seed industries as well. Dear learners, the causal agent is fungal pathogen, that is Ascochyta rabies, and the symptoms you can see on the screen. It is clearly visible how the chickpea pods get affected by these pathogens, and leaves as well also stem are affected by these pathogens, and the small circular to irregular brown lesions with the dark margins and gray centers, later it merges to form the larger blighted areas. You can see on the pods as well as on the leaves, and the stem and petioles also get affected by the elongated dark brown lesions. Sometimes it also causing the enlarge and causing the drying of the breaking of the leaves and stems also. And the pods in the circular brown spots with the black margins, it affect the pods often dry and shrivels also. And seed also get affected like a dark spot or sometime it gets shriveled conditions. And because under the humid condition the small black fruiting bodies, that is pycnodia, appears as tiny dots within the lesions. And dear learners, how this disease has caused year by year, that is disease cycle, you can say the pathogen survives in the infected seed and the plant's debris as well in the field condition and the soil condition. You can say the primary infection occurs through the infected seed as well as infected plant debris also in the soil, and secondary infection get spread through the airborne conidia during the humid weather. And rain splashes, wind and mechanical handling, it helps to the spread of the disease spores during the growing condition. The fungus produces pycnidia on the infected tissue that release the conidia which infect nearby the plants, and the sometime it completing the disease cycle and repeating the disease cycle in the field. And the favorable condition which favor the disease incidence is the disease develops rapidly during the cool weather like 15 to 25 degrees centigrade, as well as humid to cloudy conditions is favor to this disease. And how you can manage this disease by means of an integrated approach, basically combination of the resistant variety, cultural practices, biological as well as chemical method to providing the best control of ascochyta blight. Dear learners, the resistant variety, as I have told you, that like Pusa 362, JG 315, Pusa 372, so many varieties that is resistant are somehow tolerant for this ascochyta blight. And next is cultural practices. Dear learners, you have to rotate the chickpea with non-host crops such as cereals, wheat, maize or oil seed for 2 to 3 years. Field sanitation is also helpful for minimizing the disease severity, to collect and destroy the infected crop debris immediately after the harvesting to prevent the carryover of the pathogen. Use of disease free seeds, you have to use for sowing purpose basically certified pathogen free seed for sowing. Dear learners, you have to also maintain the balance for the avoiding the early sowing in areas where the prone to the cool or humid weather, and which favor the disease development basically. And you have to maintain adequate spacing between the rows to rows and plant to plant distance to avoid the circulation, to allow the circulation and reduce the humidity in the canopy. And irrigation, you have to also avoid the overhead irrigation or irrigation during the cloudy condition. And there are the biological control methods also available. You have to use the Trichoderma herzianum or Trichoderma viridi at the rate of 10 gram per kilogram seed before the sowing. The bioagent that colonizes roots and that provides the protection against soil and seed-borne inoculum. So you have to use basically seed treatment is very helpful to minimizing the pathogen load in the soil. And you have to also apply the Trichoderma herzianum at the rate of 2.5 kilogram per hectare, and you can also mix with the 25 kilogram well decomposed FYM or compost in the field before sowing. And combination with the rhizobium, you have to treat the seed or inoculated seed with the rhizobium culture to promote the nodules for long plant vigors. And the chemical is also available to treat the seed with the carbendazim or thyram at the rate of 2 to 3 gram per kilogram of the seed before sowing. And you can also spray the foliar chemicals like applies chlorothalonil or mancozeb at the rate of 2.25% after at the rate of 10 to 15 days intervals during the favorable weather for the pathogen. And you have to use 2 to 3 spray. It may be required depending on the disease severity. First spray at the disease appearance, second and third spray at the rate of 10 to 15 days interval. You have to avoid excessive use of fungicide to allow recommended dose only. Dear learners, these are the some management practices, that is IDM for Ascochyta blight of the chickpea. And now we will discuss about the powdery mildew of the pea. Dear learners, powdery mildew of the pea is also another important disease for the pulse crop. That is basically it is a common and destructive fungal disease of pea for both garden pea as well as field type of the pea, and it appears during the cool, dry weather and high humidity as well as, and it can spread rapidly at the late in the season. In India, this disease is serious in northern plains, hilly areas as well as central region during the Rabi season, particularly in the February to March month. And the disease may cause up to 20 to 50% yield loss, and under the severe condition even more than that. Late sown crop are more affected due to the favorable weather or cool nights or dry days condition. Dear learners, the symptoms for this disease, you can say basically the pathogen is a fungal pathogen, that is Erisiphe PC, and the pathogen affect the basically leaves, stem as well as pod also. So first of all, we will discuss the how symptoms appears on the leaves. Basically, the white powdery patches first appears on the lower leaves, and gradually these pathogens later spread on the upper surface of the leaves, and the patches enlarge and covering the entire leaf surface. The infected leaves turn yellow, shriveled and dry prematurely, and the symptoms on the stem and tendrils also affected by this pathogen. Similarly, white powdery fungal growth appears. You can see in the photographs how the pathogens affect the pea pods as well as leaves and tendrils also. Stem get also affected by these pathogens, and pod may also get covered with the white powders. Seed inside may be shriveled with the loss of viability, and severe infection reduce photosynthetic activity and that also get weak the plant and leading to the poor yield and to the farmers. And dear learners, disease cycle, how the pathogens circulate through year by year, and the pathogens survive as Clistothesia, that is a sexual stage of this pathogen, and in the plant debris, and as mycelium or conidia on volunteer pea plants. And primary infection get affected by the ascospores or conidia present in the field or nearby the crops, and secondary spread occurs through airborne conidia which germinate on the leaf surface, and it infect the new plants as well. Dear learners, the disease is spread rapidly in dense crop that stand and the late sown crop also, and the favorable conditions for this disease is cool weather like 20 to 25% degree centigrade, 20 to 25 degree centigrade temperature, and dry weather is favor for the spread of the disease with the relative humidity, high humidity like 60 to 80%, and little if the rainfall happen, then the disease is more severely affected to the crop. And next point is how you can manage such kind of disease, that is powdery mildew of pea. And basically host resistant, you have to first of all effective management combination to the host resistant cultural practices, biological as well as judicious use of fungicide only. And the resistant or tolerant variety available like Rachna, HFP4, AP1, JP885, Pant P5, and so many tolerant varieties or resistant varieties are available. You have to avoid susceptible cultivars in regions with the history of the disease occurrence. And the cultural practices like sowing time, and you have to early sowing like in November, it helps to avoid the favorable conditions for the disease development. You have to rotate with the non-leguminous crop like cereals or oilseed crop for 2 to 3 years to reduce the inoculum potential. And the field sanitation, you have to remove and destroy the infected crop debris and volunteer plants after the harvest. Spacing and air circulation, maintaining the optimum spacing between the row to row, and that allow to grow air movement between the plants to plant, and row to row condition. And irrigation management, you have to avoid excessive irrigation that maintain the moderate soil moisture as very humid micro limits over there, and microbial limits, and the favor to the disease spread. And you have to advise that application of balanced dose of nitrogenous fertilizer to avoid excessive growth of the vigorous growth condition to the plants. And the biological control like you have to use Trichoderma harzianum or Trichoderma viridi for the seed treatment purpose before the sowing at the rate of 10 gram per kilogram of the seed. And you have to also apply the Trichoderma harzianum 2.5 kilogram per hectare with the 25 kilogram FYM or any other compost to broadcast the field before sowing. And you can also use the foliar spray to the powdery mildew condition, and the biological spray is also available if you can apply. And dear learners, we will get a small break and we will meet again. Thank you.
जीवन ये प्रकाशित करने, राहों को आलोकित करने, हम अपना दीपक स्वयं बने हम, अपना रास्ता स्वयं चुने।
अपना दीपक स्वयं बने हम, अपना रास्ता स्वयं चुने।
जीवन ये प्रकाशित करने, राहों को आलोकित करने।
धन साधन समय की सीमा से विद्यालय जो मुक्त हो,
धन साधन समय की सीमा से विद्यालय जो मुक्त हो।
कभी पढ़े हम कहीं पढ़े, वे विषय की लोच से युक्त हो।
कभी पढ़े हम कहीं पढ़े, वे विषय की लोच से युक्त हो।
मित्रों हम उठे और जागे,
मंजिल आने की ध्वनि सुने हम। अपना दीपक स्वयं बने हम, अपना रास्ता स्वयं चुने।
जीवन ये प्रकाशित करने, राहों को आलोकित करने।
राष्ट्रीय मुक्त विद्यालय का है द्वार खुला सबकी खातिर।
शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर।
शिक्षा और परीक्षा का अवसर एक बार ही क्यों मिलता फिर। दुनिया में इसके केंद्र बात,
कोई मन चाहा केंद्र चुने हम। अपना दीपक स्वयं बने हम, अपना रास्ता स्वयं चुने। कोई मन चाहा केंद्र चुने हम। स्वयं बने हम,
Welcome back once again, dear learners. We are discussing about the major diseases of the pulse crop, and we had discussed about the Phytophthora blight, and now we will discuss about the Phytophthora blight of the pulse crop. Phytophthora blight is a serious soil-borne and water-borne disease that affect the many pulse crop such as pigeon pea, black gram, green gram, cowpea and chickpea, and it occurs mainly in high rainfall areas, and that is poorly drained soil conditions, whether the water is stagnation that favor the infection rate, and the disease cause stem rot, leaf blight and seedling damping off, and it leading to the heavy yield loss, especially during the rainy season. And the distribution, you can say it is widely distribution in India, Southeast Asia and African country, particularly in the tropical as well as subtropical region. And in the India it is common in Uttar Pradesh, Bihar, Madhya Pradesh, Maharashtra, Karnataka and Andhra Pradesh and some other parts also. And the yield loss that depends on the range between the 20 to 75%. It's depending on the crop, weather as well as management practices. And low laying and the poorly drained fields are most prone to the epidemics of this disease. And The causal organism of this disease is Phytophthora, and it is a oomycetes fungus like pathogen, and the major species that affect the pulse includes the Phytophthora drosseleri and for pigeon pea, and Phytophthora vigni in the cowpea, black gram, green gram, and Phytophthora melonis, that is generally for the chickpea and others, and it belongs to the class oomycetes, and the pathogens that produce the sporangia, zoospores and oospores that adopt for the several survival and spread of the moist soil. And basically the symptoms of this pathogen is vary slightly by crop to crop, but general features include the on seedlings. Basically first symptoms appears on the seedlings, that is damping of symptoms soon after the emergence of the crop, and seedling become water soaked near the base of the soil and get wilted and get die also. And on the stem and branches also, the water soaked dark brown lesions that near the collar region, and the lesion basically girdle the stem, leading to the sudden wilting and its collapse to the plants. And the humid weather, that is white colony fungal growth, it may the seen on the infected plants, and the on leaf and pod also the water soaked or the grassy spot appears on the leaves, and later it turn to the brown to the necrotic region, and pod also get affected and it looks like a brown patches on, and the seed inside it becomes the discolored, shriveled or rotten also, and the field appearance is also look like a patch wise affected crop quickly after the rainfall or first irrigation, and the fungus survive in the soil, and infected plants debris as well as oospore or chlamydospore. The means the disease cycle starts from the oospores survive in the infected plant debris in the soil, and the primary infection caused by the oospore that present in the soil, or it reduce residues on the previous crop, and secondary infection get started by the zoospores which move through the surface of water or rain splashes or by means of irrigation water. And mode of infection, zoospores infect the plants through the wounds or the natural openings like hydrothotes, lenticel is in the stems or in roots, and the favorable condition for the spread of the disease is 25 to 30 degree centigrade, high moistures or flooding conditions for the continuous rain or the poor drainage system that helps to the pathogen growth and development. Dear learners, how you can manage this disease in the field? Basically you have to first of all choose the resistant or somehow tolerant variety against this disease, and cultural practices, biological as well as chemical method is apply to the crop, and the host resistant, first of all the resistant tolerant variety you have to choose, and like pigeon pea and ICPL. So many varieties are available as per your requirement, you can choose out of them. And cultural practices, as I have told you, like crop rotation, you have with the help of non-leguminous crop like cereals and all seed crop for 2 to 3 years to break the disease cycle. And field sanitation, you have to remove and destroy the infected plant residues and volunteer host also, and drainage management to avoid the waterlogging condition in the field, and plant in the ridges and furrows in proper drainage area. Means you have to grow the ridge and furrows method will be the better for the managing the waterlogging condition. Dear learners, the cultural practices you have to also maintain the soil sowing time. So the early are in the recommended time to avoid the heavy rainfall. Plant spacing, you have to optimize the spacing to improve the circulation and reduce the humidity also. And soil solarization is one of the better technique you can apply with the help of transparent seed, polythene seed to 2 to 3 weeks before the sowing to reduce the inoculum level. And biological control treatment of the seed, basically with the help of Trichoderma herzianum, Trichoderma viridi at the rate of 10 gram per kilogram of the seed before sowing. And you can also apply the mix with the Trichoderma herzianum with the compost for the 7 to 10 days before the sowing. And you can dip the root with the seedling in the Trichoderma suspension for 10 gram per liter of water for 15 to 20 minutes before the sowing. And biological agent helps to reduce the pathogen that survival for the enhance the root health. And the last you can also use if the condition is not control in your hand, so that you can apply the fungicide spray like Mancozeb 0.25%, copper oxychloride 0.3% or metalaxyl 0.1% at the first sign of the disease. And you can also repeat if disease severity is not in handle, and so that within a 10 to 15 days intervals under the favorable condition. But you have to avoid excessive chemical use to prevent the resistant and residuals problem in the field. Dear learners, the next pathogen is yellow mosaic virus, that is YMV of the green gram. This is the viral pathogen, that is yellow mosaic virus is one of the most destructive viral disease of the green gram, that is Vigna radiata, and black gram also, that is Vigna mungo, and India and other tropical region. The disease causes severe yield loss due to reduced photosynthetic activity, poor pod formation and shriveled seed. It is transmitted by the white fly, that is Bemisia tabaci, to the pathogen, and distribution, as I have told you, that India, Pakistan, Bangladesh, Sri Lanka, Thailand and other part of the Southeast Asia. And in India it is so many states are affected by this disease, like Madhya Pradesh, Uttar Pradesh, Maharashtra, Punjab, Haryana, Rajasthan, Tamil Nadu and other part where the crop has been cultivating continuously, and yield loss it's very 10 to 100%, it's depending on the stages and infection of the environmental condition. And the pathogen, as I have told you, it is a viral pathogen, that is yellow mosaic virus. It's belonging to the genus Bagnovirus under the family Geminiviridae, and it is basically DNA single stranded DNA virus. And the host range is include the mung bean, urd bean, soya bean, cowpea, French bean, etc. And the symptoms, the small yellow patches appears on the young leaves, and progressive symptoms, that is patches wise it collapse, and sometimes bristol yellow, the basically mosaic pattern are on the surface of the leaves, and the leaves become thickened and brittle, and it reduced the leaf size as well as stunted the plant growth also. Infected plants produce a few flowers or pods which are often malformed with the small shriveled seed. And basically the disease cycle, primary source of inoculum Thus, the pathogens virus does not survive in the soil or seed, and the survival of the viruses during the off season is alternate host or leguminous weed, and transmission by Bemisia tabaci, that is white fly, and basically the favorable condition for the 25 to 35 degrees centigrade, and you can use the cultural management practices, timely sowing, crop rotation, and weed control also, and field sanitization is more important to manage this disease. And the chemical condition, you can also treat the you can also kill the pathogen basically which are responsible vectors, and these all about the pulse crop which get affected by the major diseases. Dear learners, hope this session is very informative to you. Thanks for precious listening and learning. Thank you very much to all dear learners. Thank you.